Synthesis from vitamin B12 and folic acid, lists the dietary sources and their requirement of vitamin B12, biochemical function of vitamin B12 and how it is digested and absorbed and transport, explain the deficiencies, other factors regulating the erythropoiesis and burn carbon metabolisms. There are different types of vitamins, water soluble and fat soluble. Water soluble non B complex ascorbic acid and B complex energy releasing hemopoietics and others. Hemopoietics include folic acid and vitamin B12. Water soluble vitamin. Water soluble vitamins not significantly stored in the body must be supplied regularly in the diet and excess in our body as excreted down. The vitamin B12 complex present in small quantity in different types of food, important for growth and good health, help in various biochemical processes in the cells, function as a coenzymes. Cobalamin, vitamin B12. Vitamin B12's main functions in healthy red blood cells and narrow nervous system functions. They also help in sperm productions, restore immune systems, improve mental energy, memory, protect against allergies and toxins. The sources are milk, egg, liver, dairy products, fish, and shellfish. Its deficiency can lead to pernicious anemia. Vitamin B12 is also called as cobalamin, cyanocobalamin, and hydroxycobalamin. It is built from nucleotides and a complex tetrapyrrole ring structure, which is called as porine ring, and a cobalt ion in the center. The R group. When R group is cyanide, the vitamin B12 is called as a cyanocobalamin. When R group is hydroxyl group, it is called as a hydroxycobalamin. And the coenzyme form of vitamin B12, the R equal as adenosyl group, it is called as adenosylcobalamin. When R is equals as a methyl, then it's called as a methylcobalamin. Coenzyme form of B12. The adenosylcobalamin and methylcobalamin is a coenzyme for metabolic reactions. Liver is a good source and dairy requirements is 2.4 microgram per day for men and women. This is the structure of vitamin B12. This is the coral ring. And when this R group is attached with cyanide, it's called cyanocobalamin. When methyl group is attached, it's called methylcobalamin. Vitamin B12 is essential for normal nervous system function and red blood cell maturation. It is not synthesized in the body, must be supplied in the diet. Will when it comes in our diet, it's first bind to the intrinsic factor and absorb into the ileum. Intrinsic factor is a protein secreted by the cell in the stomach. Absorption of vitamin B12 bind to a glycoprotein which is called an intrinsic factor in the stomach, and intrinsic factor is secreted by the parietal cell of the stomach. Vitamin and intrinsic factor complex recognize surface receptor of mucosal cell in the ileum and is absorbed there. It is stored mainly in the liver in amount 3 to 5 milligrams, sufficient to last a couple of years. Uh, increased need of vitamin B12 in pregnancy, lactation, and growth. This is the diet when take in our diet vitamin B12. It's first bind to the intrinsic factor in the parietal cell of the stomach and then goes into the ileum where it's absorbed. So as I discuss about the dietary intakes, and the stomach is mainly where the intrinsic factor synthesized, and this synthesized intrinsic factor by the parietal cells, and it goes into the duodenum. The pancreas secreted additional protease enzymes which degrade the R binder holding of the vitamin B12. Then the vitamin B12 bind to intrinsic factor for the remainder of the journey of the small intestine, and in ileum where this B complex and intrinsic factor complex passes unchanged down to the small intestine and absorb into the terminal ileums. The functions of vitamin B12, the two reactions which we have always remember. The first is the conversion of homocysteine into methionine, where vitamin B12 is essential, and the other one is the fatty acid convert into the methynyl malonyl CoA to the succinyl CoA. So these two reactions required vitamin B12, and this methynyl malonyl CoA when converted to succinyl CoA, which is the first step of synthesis of heme synthesis. So that's why the succinyl CoA is required for the heme synthesis. If vitamin B12 is not there, the heme synthesis will be affected as well. Now the homocysteine, when convert into the methionine, it's required N5-methyl tetrahydrofolate is a one form of the folic acid, and this type of folate convert into the tetrahydrofolate with the help of the methyl cobalamin or vitamin B12. This process required the methionine. This process forms actually the methionine, and if the B12 is not present, then homocystinuria occur and pernicious anemia, which is mainly due to the vitamin B12 deficiency. And absorption of B12 is mainly prevented by the intrinsic factor. If intrinsic factor is not there, so it's again the vitamin B12 deficiency occur. This is the second reactions where the methynyl malonyl CoA convert into the succinyl CoA, which is the first step of heme synthesis. So when B12 is deficient, the methyl malonic aciduria occur, and this high level of methynyl malonyl CoA is used instead of methynyl malonyl CoA for fatty acid synthesis. And the myelin sheets of our nerve neurons is required the fatty acids which is if their vitamin B12 is absent, the myelin synthesis with these abnormal fatty acids is unstable and degrade, causing the neuropathies. So B12, as we discussed, the heme synthesis is very important because sessile CoA cannot be formed, the hemoglobin cannot be formed, and vitamin B12 is also important along with the folic acid in the DNA synthesis and maturation of erythrocytes. So vitamin B12 deficiency, what is pernicious anemia? It is nothing. It is a vitamin B12 deficiency result for impaired uptake of lack of intrinsic factor, which is secreted by the parietal cell of the stomach. So, this major causes of vitamin B12 deficiency is autoimmune destruction and genetic causes, genetic defects of pernicious anemia. So, the clinical manifestation is neuropsychiatric problem occur, vomiting, heartburn, abdominal bloating, gas, diarrhea, loss of appetite, and weight loss. Mainly, we assess through the vitamin D a deficiency test, which is called a Schilling test, and check the serum intrinsic factor, intrinsic factor antibodies. Megaloblastic anemia. Vitamin B12 deficiency can also call the megaloblastic anemia because the inhibition of DNA synthesis which during the RBC production. If DNA cannot be synthesized, as a result the megaloblast and immature RBCs are formed. These RBCs have fragile membrane and get broken down more rapidly, leading to short lifespan of RBCs. The mainly how to assess the vitamin B12 deficiency. We assess the vitamin B12 deficiency to check the vitamin B12 level through ELISA and peripheral blood smear levels. Folic acid. Folic acid, the major folic acid is not biologically active, and the active coenzyme form of folic acids are tetrahydrofolate, N5-methyl tetrahydrofolate, N5 N10 methyl tetrahydrofolate, N10 formyl tetrahydrofolate, N5 formyl tetrahydrofolate. These are the major form of folic acid. Active form of folic acids are tetrahydrofolic acid. The daily requirement of folic acid is 200 to 400 microgram per day. The one carbon change of folic acids change in different forms. The N5 and 10 methyl tetrahydrofolic acid, N10 formyl tetrahydrofolic acid, N5 methyl tetrahydrofolate, and N5 N10 methylene tetrahydrofolate. These all are the active form of tetrahydrofolic or folic acid. This is the structure of folic acids where pterine rings, PABA rings, and glutamic acids they combine to form the folic acid. Folate convert into the dihydrofolate and it's convert into the tetrahydrofolate. And humans and microorganisms can form this folic acid. Folic acid is reduced within the cells, principally in the liver, where it is stored to tetrahydrofolate. These are the some active forms of tetrahydrofolates where you can see the N5 position is the site of attachment of methyl and formyl groups. The N10 is the site of attachment of formyl group and that of both for N5 and N10 bridges, the methylene and methylene groups. Folate in the foods, fortified ready to eat cereals, Dark green leafy vegetables like broccolis, orange juices, wheat, and legumes. They all contain the folic acid. Folic acid is very helpful in heme synthesis. You can see this tetrahydrofolate convert into N5, N10 methylene tetrahydrofolate mainly convert this into the glycine, which one of the another amino acid which very helpful in the synthesis of heme. So folate conversion, different folate coenzyme are specific for multiple reactions, like conversion of tryptophan into other methyl donors like purine, and the histidine is convert. These all conversion of these amino acid into the purine and pyrimidine with the help of the different active form of folate like N5 N10 methylene tetrahydrofolate, 5-methylene tetrahydrofolate, and N5 methylene tetrahydrofolate. These are some other tetrahydrofolate which actually give their methyl group to the different amino acid and form the different purines and pyrimidine. So this is called as actually the carrier of one carbon groups, which we will discuss in the later. The one carbon units are attached to or removed from it, and different metabolites change like histidine, serine, methionine, purine. Role of N5, N10 methylene tetrahydrofolate in thymine monophosphate synthesis in the most metabolic significant function for this vitamin. Vitamin B12 and N5 methylene tetrahydrofolate in the conversion of homocysteine to methionine is important in helping cell to regenerate need to tetrahydrofolate. Deficiency of folic acids mainly occur in poor dietary habits, those in chronic alcoholism, impaired absorption of metabolisms can occur the folate deficiency. Certain drugs can cause the folate deficiency. Increased demand can cause the folate deficiency, like in pregnancies. Deficiency of folate can impair the thymine monophosphate synthesis and purine synthesis, which can result into a megablastic anemia. And these are the pictures of neural tube defects, which is due to the deficiency of folate in pregnancy. Folate deficiency can also can causes the macrocytic anemia. What is macrocytic anemia? During the RBC generation, DNA synthesis is delayed, but protein synthesis is continuous, and hemoglobin accumulate and RBC precursor forms. And the ultimately immature nucleus and mature eosinophil cytoplasm in bone marrow, which and we can see in this type of deficiency the reticulocytosis is seen, and hemolysis is occurred due to the rapid destruction of RBCs in the spleen, leading to short half-life. We check the serum folate levels, and normal folic acid level in serum is about 20 nanogram per ml and measured by the radioimmunoassay, and peripheral picture we can see the macrocytic hypochromic anemia. Regulation of erythropoiesis are very important, and the multiple factors affecting the erythropoiesis, tissue oxygenation like erythropoietins, and the vitamins is very important for the regulation of erythropoiesis, as we discussed earlier about the vitamin B12 and the folic acids. So there are other vitamins also help, the metals, proteins, hormones. These are the some vitamins which help in the hemopoiesis, like B12 and folic acid, which we have already discussed. But B6, vitamin C, and vitamin B5 also help in the hemopoiesis. One carbon metabolism is nothing, it's just the giving the methyl group by the folate and vitamin B12 to the other compound and change to form the to form the glycine, serine, or succinyl CoA. Similarly, when folate form convert into a tetrahydrofolate, and it's ultimately converted to the 5,10 methylene tetrahydrofolate, which is helpful in the nucleic acid synthesis. And this 5-methylene tetrahydrofolate again help in the homocysteine to convert into the methionine. Methionine is very important carbon donor in our body in the form of as SAM. SAM is nothing as adenosyl methionine.
is called as SAM, which gives the methyl group to other multiple reaction in our bodies to give their methyl group. So this shows how the dihydrofolate is converted dihydrofolate into tetrahydrofolate. As I told you, this tetrahydrofolate can further convert into the active form N5 and 10 methylene, which is used in purine pyrimidine synthesis. And this tetrahydrofolate also help in the homocysteine to form methionine, and methionine form the S-adenosyl methyl.